Photograph a historic site, help Wikipedia, and win a prize. Participate in the world's largest photography competition this month. Learn more. Vault, organelle. 11 languages. Show. Vault, N-terminal repeat domain. Structure of the vault complex from rat liver. 1. Identifiers. Symbol vault. FAM PF01505. Interpro IPR 002499. ProZit PTC 51224. Available protein structures. The vault or vault cytoplasmic ribonucleoprotein is a eukaryotic organelle whose function is not yet fully understood. Discovered and isolated by Nancy Kedeshar and Leonard Rome in 1986. 2. Vaults are cytoplasmic organelles which, when negative stained and viewed under an electron microscope, resemble the arches of a cathedral's vaulted ceiling, with 39-fold, or D. 39D, symmetry. 1. They are present in many types of eukaryotic cells, and appear to be highly conserved among eukaryotes. 3. Morphology, edit. Vaults are large ribonucleoprotein particles, about 3 times the size of a ribosome and weighing approximately 13 m. They are found in most eukaryotic cells and all higher eukaryotes. They measure 34 nm by 60 nm from a negative stain, 26 nm by 49 nm from cryo-electron microscopy, and 35 nm by 59 nm from STEM. 4. The vaults consist primarily of proteins, making it difficult to stain with conventional techniques. Structure, edit. The protein structure consists of an outer shell composed of 78 copies of the -100K, the major vault protein, MVP. Inside are two associated vault proteins, TEP1 and VPAP. TEP1, also known as the telomerase-associated protein 1, 5, is 290K, and VPAP, also known as PAP4, is related to poly-ADP ribose, polymerase, PAP, and is 193K. 6. Vaults from higher eukaryotes also contain one or several small vault RNAs, VNAs, also known as VDNAs, of 86141 bases within. 7. The MVP subunits are composed head-to-head, with the N-termini of each half-vault facing each other. From the N-terminal to the C-terminal, a MVP subunit folds into 9 repeat domains, 1 band 7. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Like shoulder domain, 1 cap helix domain, and 1 cap ring domain, corresponding to the shape of the vault shell. VPAP binds to repeat domain #4. TEP1, itself a ring due to the WD40 repeat, binds to the cap domain, with one particular type of vRNA plugging the cap. 8. Function, edit. Despite not being fully elucidated, vaults have been associated with the nuclear pore complexes, and their octagonal shape appears to support this. 9. 10. Vaults have been implicated in a broad range of cellular functions including nuclear cytoplasmic transport, mRNA localization, drug resistance, cell signaling, nuclear pore assembly, and innate immunity. 11. The 3 vault proteins MVP, VPAP, and TEP1 have each been knocked out individually and in combination, VPAP and TEP1, in mice. 12, 13, 14. All of the knockout mice are viable and no major phenotypic alterations have been observed. Dictyostelium encode 3 different MVPs, 2 of which have been knocked out singly and in combination. 15. The only phenotype seen in the Dictyostelium double knockout was growth retardation under nutritional stress. 16. If volts are involved in essential cellular functions, it seems likely that redundant systems exist that can ameliorate their loss. Association with cancer, edit. In the late 1990s, researchers found that volts, especially the MVP, were overexpressed in cancer patients who were diagnosed with multidrug resistance, that is the resistance against many chemotherapy treatments. 17. Although this does not prove that increased number of volts led to drug resistance, it does hint at some sort of involvement. This has potential in discovering the mechanisms behind drug resistance in tumor cells and improving anti-cancer drugs. 15. Evolutionary conservation, edit. Volts have been identified in mammals, amphibians, avians and Dictyostelium discoideum. 3. The volt model used by the FAM database identifies homologs in Paramecium tetraeolia, Kinetoplastida, many vertebrates, a cnidarian, starlet sea anemone, mollusks, trichoplax, adherens, flateworms, Echinococcus granulosus and choanoflagellate. 18. Although volts have been observed in many eukaryotic species, a few species do not appear to have the ribonucleoprotein. These include, 19. Arabidopsis thaliana, a small flowering plant related to cabbage and mustard. Caenorhabditis elegans, a free-living nematode that lives in soil. Drosophila melanogaster, a two-winged insect also known as a fruit fly. Saccharomyces cerevisiae, a species of yeast. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. These 4 species are model organisms for plants, nematodes, animal genetics and fungi, respectively. Despite these exceptions, the high degree of similarity of volts in organisms that do have them implies some sort of evolutionary importance. 3. Homologs of the major volt protein has been computationally found in bacteria. Cyanobacterial sequences appear most similar. 20. 21. FAM is also able to identify some such homologs. 18. Volt engineering, edit. The Rome lab at UCLA has collaborated with a number of groups to use the baculovirus system to produce large quantities of volts. When the major volt protein, MVP, is expressed in insect cells, volt particles are assembled on polyribosums in the cytoplasm. 22. By using molecular genetic techniques to modify the gene encoding the major volt protein, volt particles have been produced with chemically active peptides attached to their sequence. These modified proteins are incorporated into the inside of the volt particle without altering its basic structure. Proteins and peptides can also be packaged into volts by attachment of a packaging domain derived from the VBARP protein. 16. A number of modified volt particles have been produced in order to test the concept that volts can be bioengineered to allow their use in a wide variety of biological applications including drug delivery, biological sensors, enzyme delivery, controlled release, and environmental remediation. A volt has been packaged with a chemokine for potential use to activate the immune system to attack lung cancer, and this approach has undergone phase I trials. 23, 24. See also, edit. Volt RNA. Major Volt protein. References, edit. 1 A beta NACAR H, Kato K, Yamashita E, Sumizawa T, Ju Y, Yao M, Iwasaki K, Yoshimura M, Zukihara T, January 2009. The structure of rat liver volt at 3.5 angstrom resolution. Science. 323, 5912. 3848. Bibcode, 2009 SCI, 323384T. Do, 10.1116/science.11649775. PMID 191508446. S2 SID 2072790. 2 Kedashar NL, Rome LH, September 1986. Isolation and characterization of a novel ribonucleoprotein particle. Large structures contain a single species of small RNA. The Journal of Cell Biology. 103, 3699709 do. 10.1083/JCB.103.3.699. PMC 21140306. PMID 29437444. 3 ABC Kedashar NL, Meikle MC, Bittner D, Rome LH, April 1990. Volts. 2. Ribonucleoprotein. Structures are highly conserved among higher and lower eukaryotes. The Journal of Cell Biology. 110, 4895901 do. 10.1083/JCB.110.4.895. PMC 21161006. PMID 1691193. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 4 Kedashar NL, Huser JE, Chugani DC, Rome LH, January 1991. Volts. 3. Volt ribonucleoprotein. Particles open into flower-like structures with octagonal symmetry. The Journal of Cell Biology. 112. 222535 do. 10.1083/JCB.112.2.225. PMC 228884. PMID 1988458. 5 Kikuifa VA, Stephen A.G., Harrington L., Robinson Mo., Rome LH, November 1999. Volts and telomerase share a common subunit, TEP1. The Journal of Biological Chemistry. 274, 4632712. 7 do. 10.1074/JBC.274.46.32712. PMID 10551828. 6 Kikuifa VA, Siva AC, Kedashar NL, Inman M, Ruland C, Struli M, Rome LH, September 1999. The 193Kd Volt protein, VPOP, is a novel poly-ADP ribose polymerase. The Journal of Cell Biology. 146, 591728 do. 10.1083/JCB.146.5.917. PMC 21699495. PMID 1047748. 7 Van Zonne, Mosin Chem H, Skipper RJ, Sonneveld P, Weimer EA, September 2003. The Volt. Complex. Cellular and Molecular Life Sciences. 60, 9/18/2837 do. 10.1007/00018-003-3030 y. PMC 111388885. PMID 145235546. S2 SID 211962662. 8 Tanaka, Hideki, Tsukihara, Tomitake, 2012. Structural Studies of Large Nucleoprotein Particles. Volts. Proceedings of the Japan Academy, Series B88, 8416433. Bibcode, 2012 PUB, 88416T. Do. 10.2183/PUB.88.416. PMC 3491081. PMID 230602231. 9 Chugani DC, Rome LH, Kedashar NL, September 1993. Evidence that volt-ribonucleoprotein particles localize to the nuclear pore complex. Journal of Cell Science. 106, pt 123-9. Do. 10.1042/JCS.106.1.23. PMID 827027. 10 Unwin PN, Milligan Ra, April 1982. A large particle associated with the perimeter of the nuclear pore complex. The Journal of Cell Biology. 93, 16375 do. 10.1083/JCB.93.1.63. PMC 211207. PMID 7068761. 11 Berger W., Steiner E., Grusk M., Elberling L., Mick January 2009. Volts and the Major Volt. Protein, Novel Roles in Signal Pathway Regulation and Immunity. Cellular and Molecular Life Sciences. 66, 14361 do. 10.1007/008-008-8364Z. PMC 11131553. PMID 187591128. S2 SID 6326163. 12 Kikuifa VA, Lu Y, Compound, Snow B, Stewart P.L., Harrington L., Rome L.H., January 2001. The telomerase /volt-associated protein TEP1 is required for volt RNA stability and its association with the volt particle. The Journal of Cell Biology. 152, 115764 do. 10.1083/JCB.152.1.157. PMC 21936551. PMID 11149928. 13 Lu Y, Snow B, Han DMP, Bailosher G, Kikuifa VA, Young D, Wakamo, ITO, Siderovsky. DP, Lansdorp PM, Robinson Mo, Harrington L., November 2000. Telomerase-associated protein TEP1 is not essential for telomerase activity or telomere length maintenance in vivo. Molecular and cellular biology. 2021 8178 do. 10.1128/MCB.20.21.8178-8184.2000. PMC 86427. PMID 1102727. 14 carat Mosin MH, Van Zonne, Franzel Lutani, Skoester M, Kikhuifa VA, Skiffer GL, Skipa RJ. Sonneveld P, Weema EA, December 2002. Disruption of the murine major volt protein, MVP/LRP. Gene does not induce hypersensitivity to cytostatics. Cancer Research. 62, 24 7298 304. PMID 1249927. 15 carat AB Kikhuifa VA, Vaju SK, Rome LH, May 1996. Volts are the answer, what is the question? Trends in Cell Biology. 6, 51748 10.1016/0962-8924, 96, 10014-3. PMID 1515748. 16 carat AB Rome LH, Kikhuifa VA, February 2013. Development of the volt particle as a platform. Technology. ACS Nano. 7, 2889902 10.1021/NN305208. PMID 232676774. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 17 carat Mosin MH, Van Zonne, Skipa RJ, Sonneveld P, Weema EA, October 2003. Volts, a ribonucleoprotein particle involved in drug resistance. Oncogen. 22, 47 7458 67. Do. 10.1038/sj.onc.120697. PMID 1457685. 18 carat AB http://fam.xfam.org/family/pf01505 Major Volt Protein Repeat Fam Family. 19 carat Rome L, Kedeshar N, Chugani D, August 1991. Unlocking Volts, Organelles in Search of a Function. Trends in Cell Biology. 1, 2, 3, 47, 52, 10.1016/0962-8924, 91, 90088Q. PMID 1473165. 20 carat Daily, TK, Sutherland Smith, Aj, Penny, D, 2013. In silico resurrection of the major volt protein. Suggests it is ancestral in modern eukaryotes. Genome Biology and Evolution. 5, 8 156783. Do. 10.1093/GBE/EVT113. PMC 3762200. PMID 2388792. 21 carat Sokolsky, Timofei, the 12th of December 2019. Bacterial major volt protein homologs shed new light on origins of the enigmatic organelle. Bioexif 10.1101/872010. 22 carat Mrazik J, Toso D, Ryazantsev S, Chang X, Ju ZH, Fernandez BC, Kikhuifa VA, Rome LH. November 2014. Polyribosms are molecular 3D nanoprinters that orchestrate the assembly of volt particles. ACS Nano. 8, 11115529 do, 10.1021/NN504778H. PMC 4245718. PMID 25354757. 23 carat Shoma S, Chul, Srivastava MK, Harris White M, Huang M, Li J M, Rosen F, Li G, Wang G. Kikhuifa V, Rome LH, Baratelli F, St. John M, Recomp K, Chulyong S, Hillinger S, Striata R. Dubinit S, January 2013. CCL21 Chemokine Therapy for Lung Cancer. International Trends in Immunity. 1, 11015. PMC 4175527. PMID 25264541. 24 carat Car UK, Srivastava MK, Anderson O, Baratelli F, Huang M, Kikhuifa VA, Dubinit SM, Rome LH. Shoma S, May 2011. Novel CCL 21-volt nanocapsule intratumoral delivery inhibits lung cancer growth. PLOS 1. 6, 5E18758. Bib code 2011 PLOSO 618758K. Do 10.1371/journal.pwn.0018758. PMC 3086906. PMID 21559281. External links, edit. Vault website, UCLA. Web archive, the 28th of February 2009. Vault and ribonucleoprotein and particles at the U.S. National Library of Medicine. Medical subject. Headings, Mesh. Page 4 Volt RNA at AFAM. Major Volt protein on Proteopedia. Categories, ribonucleoproteins, organelles. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.